Hello friends, Dr. Mustafa is here with your brand new class, online class of medical biotechnology. And I have added a course content to this course uh, since I did this because I can do so. I have added coronavirus pandemic as one of the topics you should learn in the medical biotechnology course. So we will go from here directly to our slides. And then I will be talking about the different aspects of coronavirus and the disease particularly, and the diagnosis and treatment options we are having. And I want you people that this is not everything which I'm telling you. And of course, that we can learn more and more, and you people can, I believe you people more than on my wisdom, then you can browse around and you can see and study papers and can share your knowledge with the class and with me in the meanwhile as well. Okay, so we will be talking about the coronavirus and the pandemic COVID-19. And before that, I wish all of you that I really hoping and I'm wishing that all of you are having and trying your level best, not only to protect yourself, but also you are working to protect your friends and family from the disease at this time. Okay, so here we go. So I will be sharing my screen with you people, so we can go ahead in a way we should. Okay, so here we go. First, I will going to talk about the disease. So this is our first lecture. The lecture is already loaded on your Moodle as well as on the Google Classroom, and the associated websites and certain other resources I have added to the places which I mentioned in my earlier video and lectures. And I will be updating these things as well. And whatever the updates I will be making with this topic, I will let you know through the means we are having to communicate with each other. Okay. Remember that if you want me to, if you want to have an interactive session with me, we will assign a particular time for that thing as well. So in that case, I can listen from you, and I will be answering your questions as well. And of course, I need that session to make assessments about you, that whether you are learning in a way I want you or not. Okay, so here we go. So in this lecture, I will be talking about the coronavirus, its structure, diagnosis, and treatment options. So first, you need to know about the viruses. Actually, you people know a lot of things, maybe more than me, about the viruses and its structure. And you know, there are two types of viruses. The one which we call as the DNA viruses, like hepatitis B, and the other they are called as RNA viruses, like hepatitis C, HIV, and this one, the coronavirus. Remember, coronavirus is the name of the family. It has many members, and some of the members were very famous earlier, like the SARS virus and the MERS virus. And most of these viruses usually uh, uh, found to be related with an intermediate member, a host member. For example, sometimes it was a bird out there, sometimes it was a pig out there, sometimes it was like a camel, which is acting as a intermediate member and transferring the virus from one species to humans. So, but in this case, we are still struggling, and we are trying, we means the scientific community is trying to figure out that who is the actual real source behind the present coronavirus. Because when it was appeared in Wuhan at the end of the 2019, the Chinese scientists shared the genetical structure, the genome sequence of that virus, and they just came to know about that thing, that it has nothing much similar to the earlier strains of the SARS and the other viruses, though they just started naming it as a novel coronavirus after that. And this particular virus is the RNA virus, so this is a little bit tricky thing as well for the viruses and for the humans as well, because dealing with RNA is a little bit difficult. Dealing with RNA based viruses are a little bit difficult as compared to the other viruses who are carrying DNA. So like any other virus, it has a protein code structure. containing glycoproteins and different proteins, and enclosed in its genetical material, which is an RNA. Usually, viruses are considered as not considered as living like other bacteria and other germs. And you know why? Because virus contains the minimum stuff which is required to exist as a living thing. I mean, it has a nucleic acids and it has contains proteins. But once these things are present outside the living system, the virus is dead. So virus is dead when it is present outside the host cell. But once the virus managed to find its place within the living system, within the host cell, it started using the machinery of the host cell. It started using the translational machinery of the host cell and start producing the things of its choice. It actually hijacked the host genetical machinery and start making use of that machinery to produce the things which belongs to the virus. So that is the case most of the time. And since I told you that virus require a host, so which of the virus is related to which particular disease? It depends on the nature and the choice of the host. For example, if a virus is selecting liver cells as a host, if it is compatible to use the liver cells as a host, then it will start affecting the liver cells like hepatitis B and C. But if the virus is optimized to use the cells present in the lungs, so in that case, it can start affecting the lungs and can result in pneumonia and the other respiratory diseases. So coronavirus is designed to affect the particular cells present within the lungs. Actually, lungs are made up of different things, and one type of cells are responsible to create the air tracts. And the novel coronavirus can associate themselves with those cells which are constituting the air tracts. So that's why you know that the air and the air tracts, they are the air present outside the body and the air present within the air tract. There is no barrier between these two things. So whatever is present in the outside air is also present right within the air tract. So the access point is very easy. Imagine in case of the hepatitis B and C virus, the virus needs to reach to the liver. It needs to pass on through different layers, to different other tissues as well. So it spends a lot of time before actually it managed to find its target. So the immune system also managed to find enough time to deal with these type of viruses. But in this case, the virus is just there and the host cell is just there as well. So the nature, the intensity of infection, the probability and chances of infection is much, much higher in this particular case. So first, when the Chinese scientists discovered that this is a new thing, this is not very much similar with the other viruses, they named it as the novel coronavirus, or like SARS-CoV-2, because most of the genome is similar to the SARS virus, but it is different. So you need to go to that link to find that paper and try to study that paper. So I just highlighted some of the things, that it has 79% nucleotide identity with the SARS-CoV, which is the original virus, the SARS virus earlier, and it has 51.8% identity with the MERS. And this difference is hell of the difference. You cannot consider this particular virus as SARS-CoV or MERS-CoV. That is why it is considered as novel. And excuse me. And also it has 87.7% identity with another virus which is present in the bat. So this is the only evidence we are having right now, which is indicating that this virus is actually originated from the bat. And you might remember the very famous movie, The Contagion. That movie, the flu in that particular movie was also originated from that bat as well. And also the choice of a virus depends on its proteins which the virus is using to bind itself with the host receptors or the host cells. And the amino acid sequence also, remember the receptor binding domain here, the receptor binding domain, this one, it also resembles with the virus which which was found to be present in the bats as well. But there are other candidate animals as well, certain reptiles and the pangolin as well, which has a similar sequence of coronaviruses as compared to this one, the novel coronavirus, the SARS-CoV-2. Okay, so just browse that paper to get more of the similarity understanding. The next thing you need to know about this virus, particularly the pandemic, the reason of pandemic, the coronavirus, you need to know about certain important terms. The first term you should know is about the virus ability to replicate itself, or virus ability to infect other people. So it is called as the R0. It's not R0, but it is like R0. It's called the replication rate. If a virus has a replication rate of 2, that means it can affect 2 people on average. And you can guess that what will happen after that, after 10 or 15 or 20 replications, you will end up with hundreds of thousands of people after that. But in case of coronavirus, COVID-19, the R0 was calculated by these people. The information is present in this article. So a lot of things you need to go and browse and see the articles. So I tried to extract the information, important information out of the articles for you people, but I'm expecting you to go and see whatever I'm telling you is actually true or not. So it's about 2.24 to 3.58. The R0 was estimated. It's quite high. It's pretty high. I mean, it's very dangerous virus that it can affect like a lot of people. And that is why the social distancing is something which everybody is talking about nowadays. Because by social distancing, we can eliminate the intermediate members out of this chain reaction, and we can reduce the possibility of having infected people out there. That's why it is important to know. The next thing is the term. I want you to be very clear about the different terms which people are using out there in the social media and also in the news, because these are quite messy information, and everybody is an expert. I mean, I'm not an expert as well, but somehow we need to study a little bit more about the terms, what that exactly mean. When the people are saying that they are screening people for coronavirus and the pandemic, screening is not a test, a diagnostic test. It's something like looking at the person. It's something like asking about the person, how you are feeling. For example, excuse me, right now I'm having a cough. So you're asking somebody, do you have a cough? Excuse me. You are analyzing or checking the temperature of the person, that whether the person is having a fever or not. So these are the initial symptoms, initial reasons to suspect a person, that whether that person might be, but of course this is not all. The fever can be because of so many reasons, and one of the reason is but this is like initial stuff. Screening is not something which you can take as a confirmation that somebody is carrying the coronavirus or not. So when people are screened on the airport, that does not mean if somebody is not having a fever that the person will not going to show the symptoms of coronavirus after that. Don't scare. Okay. Next. What are the diagnostic options we are having in order to test any virus, particularly the coronavirus? So usually a virus can be detected with the 2 types of live components present in them. The first is the virus contains certain proteins. So we can use antibodies to detect the presence of these proteins and to detect the presence and to detect the virus. The second thing is that we can use the information present in the nucleic acid of the virus. So we can detect the protein, or we can detect the nucleic acids present in the virus. So in this case, right now, the people are working very hard to analyze and to understand the proteins present in the virus coat. So they can create an antibodies against those proteins, and they can develop a diagnostic kit like the other diagnostic kits we are having. If you can remember, the pregnancy test is done with the help of a strip, and that strip contains an antibody, and when a particular protein is produced within the urine after the pregnancy, that can be tested with the help of that strip. Very common and very famous diagnostic tool people are using. So first we need to know what? We need to know about the structure of the protein. But remember, many of the proteins are similar to the other viruses, like the flu viruses. So we need to know one specific protein present in the coronavirus, the novel coronavirus, so that we can design a particular tool. We can design an antibody. And you know what we are using? We are using Eliza for that purpose. That we can develop Eliza kit for that purpose. So there's different type of Eliza options are out there, but they are dependent on that knowledge that we know a specific protein which is specifically produced in the novel coronavirus. If we don't know that information, we cannot design that kit at all. So whatever the companies in the world right now which are trying to create a diagnostic kit, they are investigating a specific protein. They are trying to find out a specific protein combination in the coronavirus, which they can use as a lead, and then they can create antibodies against it, and then they design the Eliza. The Eliza is very simple, you know, but I'm just going through one of the most specific form of Eliza, which is called as sandwich Eliza, in which we use a capture antibody. So this capture antibody is designed is something which are created in a way that it can specifically bind one particular protein, and you can guess this protein belongs to the virus. It's a specific protein present only in novel coronavirus. We don't know yet. Okay, we are working. People might be very close. People might be having it, but in the news and in the other people, we don't know one particular protein which can specifically bind it. So I believe that it is there. Almost like we are almost there to have an antibody which can specifically bind with a specific protein present in the novel coronavirus. After that, we use another antibody which can bind to that specific target molecule of the coronavirus. And then the secondary antibody which are chemically linked with an enzyme. The green one is an enzyme. And the chemical linkage is usually created with the help of biotin streptavidin combination. So these are like a chemical link which connects this enzyme with the secondary antibody, which binds with the primary antibody, which is specific to a protein present in the virus. So let's suppose if the sample contains that protein will stick to the capture antibody, and we supply this complex in a sequence. First, we provide the primary antibody. If this stuff is there, the primary antibody will stuck. If the stuff is not there, the primary antibody will be washed away. And then we provide the secondary antibody to the primary one along with the enzyme. So after all this thing, we execute a chemical reaction which produces a color. This is a substrate of this enzyme. It produces a color, and that color can be detected using a photometer or using a particular instrument. The color indicates the presence and activity of that enzyme, which indirectly means the presence of that specific protein, and the person will be considered as infected with the novel coronavirus. That's one way. The other way to detect a particular virus is to detect its nucleic acid. Now the coronavirus contains RNA. So we cannot detect the RNA directly, although there exist many ways to detect the RNA directly. But the amount is too less to be detected from a sample. So since it's not a DNA, so we cannot amplify the RNA directly. So when we extract the sample from the patients who are containing the RNA virus like HIV or HCV, or now the coronavirus patient, we extract the blood, we get the blood, and then we extract the RNA out of that blood. If the coronavirus is there, the RNA will be extracted. It will be converted into cDNA. The RNA will be converted into cDNA using the reverse transcriptase enzyme. And once the cDNA is there, we can use specific primers to amplify that cDNA. So if the product or amplification is done, we can confirm the presence of coronavirus RNA in that sample. This is called the PCR test for the coronavirus detection. And this is the only tool we are having right now in Pakistan. Whenever you heard about the things that we need that many kits, those kits means the chemicals required to execute the PCR reaction to detect the presence of coronavirus. So this is a sample picture that how these experiments are done. Usually we have in the lane number 1, we have a positive control. In the lane number 2, we have a negative control. I mean, we have a specific DNA, which is similar to the coronavirus. We have another DNA, which is not similar to the coronavirus. And then 3 and 4 are the samples from the patients. So if the patients give us the amplification product after all that process, we can refer those patients infected and contains the coronavirus. So what are the things which are required to execute this experiment? I mean, as a medical biotechnology student, you need to think that whether you can create that kit or not. Or what are the things you need to know? The first thing you need to know is to know about the sequence of the RNA in coronavirus. That's all, the sequence. So do we have the sequence? The answer is yes. Who gave this sequence to us? I shared you with the paper. The Chinese originally shared that sequence with us. And now every other day, the people are analyzing the sequence of different strains of coronavirus from different countries. So we have the sequence of the RNA. That means we can convert them into cDNA. We know the sequence that we can design the primers, which can specifically amplify a portion from that nucleic acid of virus to confirm the presence of the coronavirus in the patient. So when you heard the news, I don't want to be offensive, but in news and in other channels, when you just heard the news that somebody has developed the kit to detect the coronavirus, I don't want to mention the name, you people. I mean, it's right or wrong, but somebody's trying. I like the effort, but you should understand as a student what those people have done. उन्होंने क्या किया है? What they have done is that they have created the primers which are specific to the coronavirus nucleic acid sequence. So they can amplify. Once they extracted the RNA, they can convert it into cDNA, and after that they can amplify. They have created the primers. So that primers tubes, which are shown in the media and other places, they are the primer tubes, which you can use in the PCR machine in your School of Life Sciences laboratory and other places, those primers to amplify, to confirm the presence and absence of viral genome. So this is just the primer designing for the, we did not create, we did not find out the sequence. We just designed the primers based on the sequence. And that's okay. I mean, that's fine. For some people, somebody is doing something rather than doing nothing. So this is something which you need to know. So you can create a kit. All you need to know is how to design those primers. By the way, I asked you at the start that I will teach you all the complication, all the things, and I will be teaching you how to design primers in the upcoming lecture as well. So you can have your own coronavirus kit. You can also propose, and you can be a star on the nowadays. With somebody don't understand that what you have created, so it will highlight you more than you actually are. So these are things you need to know. If it is that much easy, I mean, just designing the primer and then creating a PCR reaction, why we are so worried about detecting the infection out there? Why we are running short of the kits? Why we are depending on the other country for the kits? Because this is a very valid question. If it is that much simple, why not we are all having PCR reactions going on? Why we stop the universities to university laboratories? We can execute those reactions on daily basis, so we can confirm the presence of coronavirus genome in any sample using the PCR machines present in our labs as well. But the answer is not that simple. It's quite complicated. Let me explain you why. Because in order to deal with the infectious sample, you need to know, you need to have labs which are equipped with a very high standard of biosafety. They are called as biosafety levels 1, 2, 3, and 4. And I want you people to go and to study what that means when I said that you need a lab of with the biosafety level 3, or you need a lab with the biosafety level 4. Since we don't know, we don't have an antiviral drug, we don't have a treatment, we don't have a solution. So we can't risk people. If we ask people to come at one place to give away their samples for the analysis, that means we should have a place where we can deal with those samples in a very, in a very protective way. So we need to have we don't have those labs. I mean, we have just 2 labs, I believe, which has the status of biosafety level 3. I guess one is in Islamabad and another one is Karachi, but you can increase my knowledge if there are more labs out there. So this is the problem. So it's not about creating the primers only, it's about how you can execute that, that stuff. So whatever the government is doing right now, the government is creating like an isolated places where people can give their samples, and those samples will be used, and RNA was extracted, cDNA was made, and then using those primers, they amplified the specific region of the coronavirus. So you can guess, you need a very, very good quality chemicals to extract the RNA first, so that there should not be any other XYZ things out there. You need to have a very accurate cDNA synthesis chemicals. Otherwise, you will start having a false positive results. And failure in this case is a big trouble. I mean, if you just analyze somebody for a particular disease like common flu or other thing using the PCR reaction, we don't do this basically. We have a solution for the common flu. But if you prove to be wrong in your analysis, that's fine. It will not hurt much to the society. But if you are not correct in analyzing the coronavirus, and you just said somebody that he is not infected, and if you have a false positive results or false negative result, that will create a chaos in the society after that. So you need to be very accurate, and you need to have a very good pair of hands for doing all those things. Okay. So what are the treatment options we are having? So this is the first part of the lecture. You can take a break, and after that I will be talking that how we can actually what are the treatment options we are having to deal with the coronavirus. Okay, this is the first part. Let's take a break.